Hello, everyone. Thank you for joining us for today's webinar, Immune Secrets of the Lymph Node Revealed by Spatial Multiomics. My name is Elizabeth Schneider, Senior Global Marketing Manager at NanoString, and I'll be your moderator for today's event. Today's educational webinar is presented by LabRoots and brought to you by those of us at NanoString. To learn more, you can visit nanostring.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues you're experiencing here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker from NanoString, Jason Reeves, who's Associate Director of Computational Biology. A little bit about Jason. So Jason is responsible, he and his team is responsible for developing analytical solutions for NanoString's Technology Access Program for the GeoMx Digital Spatial Profiler, as well as the CosMx Spatial Molecular Imager, as well as data analysis services that support academic and industry partnerships across oncology, rare and infectious disease, and neurobiology. So thank you so much, Jason, for joining us today, and we are looking forward to your presentation.
Thanks, Elizabeth, for the introduction, and it's great to be here to talk to everybody today. I hope that everybody is having a fantastic week and that we can give you a little bit of insight as to how we approach some experimental design when we want to understand the biologies of complex tissue structures, and give you a little bit of biology about what's happening within the lymph node itself during various different states and transitions that happen within that system. As Elizabeth mentioned, if you have questions, please feel free to drop those into the question Q&A box. I'll look forward to answering those, and I will try to do so both either in context or at the end of the session. So thank you again for joining us today. Now, I wanted to start with just a little bit of background on the lymphatic system and some of the reasons why we want to understand the lymph node in general and what its role is in biology. The lymphatic system is one of the systems that connects the whole body that really helps us monitor and defend ourselves against pathogens, as well as regulate the development of various different organs and prevent cancer from forming. The immune system has had an increasingly recognized role across the development of many different disease states, and the lymph node itself is a particular organ structure that develops in many different locations. So it's not something that is specific to one particular spot within the body. You've got them all over. You've got them under your chin, under your armpits. And what these areas do is allow local signals of detection of particular antigens to be transmitted throughout the body so that when there's a particular need to mobilize the immune system, you can actually do that. The lymph node as an organ itself is complex in structure, but is arrayed in a way that creates patterns of types of regions within the tissue for different particular functions. As you are closer to the edge of the outside, you're in this region called the cortex. And within that is where T cells, B cells, and myeloid cells come together to interact and identify which cells should mature to become active immune cells. As you move further into the structure of the lymph node, you move into what's called the medulla. And this structure helps organize the overall tissue architecture as well as helps facilitate transfer throughout the body as it connects to lymphatic draining vessels. The lymph node is a host for pretty much every immune cell type within the body. There's a handful that are tissue resident that never really come into contact with the lymph node, but these serve as hubs for developing immunity. And so they as I mentioned before, play a critical role in how various different diseases are regulated, but not just viral and bacterial infections. They really do impact how many different diseases develop. And there's growing evidence, even in neurobiology, of how the lymph node has direct interplay with the nervous system. So when we think about how to study this tissue, we recognize that there's a couple different components that we need to understand the actual functions that are being performed here, as well as how cells mature. The first is that this organ is composed of both key cell types that are going to be moving throughout the tissue, as well as different structures that are important to focus on, and that you can only really retain if you're doing an approach that is spatially resolved. Now, that particular aspect is something that has come into play over the last couple of years. And just as a quick history tour of the ways in which we've had different technologies for exploring transcriptomics or proteomics, it's important to take a step back to understand the foundations that have been laid within the system. Back in the early 2000s was when we first were able to start actually sequencing the whole transcriptome. And this gives you the idea that, we can understand disease states versus non-disease states, but all we're getting is a snapshot picture of all the cells and all the activation states mashed together. It's better than not having anything, but it's really coarse in terms of what you're getting out of that in terms of information. And here, this is lifted and adapted from Aviv Regev's slide, where she's describing this as basically drinking a smoothie. You got a sense of all the components that are in there, but you don't really know all the different pieces of it. And there might be some that you underappreciate because they're not very common. Next, the next big jump was in the early 2010s, and this was when single-cell sequencing became robust and feasible for most folks. And this now allows you, instead of drinking a smoothie, you now actually understand which fruits went into it. But single-cell sequencing on its own does have some biases, and it is not always possible to capture every single cell type that's present within a tissue. Moreover, it's also not possible to really know whether or not the oranges are supposed to be sitting next to the apples in your actual tissue, or if what you're looking at is really representative of the structure as a whole. And so that's where spatial biology really comes in. We've created both the GeoMx digital spatial profiler and the CosMx spatial molecular imager to now take all the components that we have and capture a snapshot of where they sit within the tissue. I'm going to tell you a little bit more about these, but I'm also going to walk you through the tissues as we go through this. So as I mentioned, GeoMx and CosMx serve different roles in terms of trying to identify and profile transcriptomic and proteomic profiles from tissues. GeoMx is a multicellular profiling approach where you can look at different tissue types or different cell types as they are spatially resolved within a tissue. We'll go through this as our first look into the lymph node itself. And then CosMx is a subcellular platform where you're getting spatial resolution of thousands of transcripts at once down to the individual cell. And that'll be described a little bit more towards the end of the talk. So I want to talk through how GeoMx works in case you aren't familiar. GeoMx combines a couple different features to really create the magic that we have with this platform. The first is that with both of our platforms, you get a 4-channel multiplex immunofluorescent stain. So you have an ability to physically look at your tissue. And I'll come into this for the lymph node in just a second. It also uses two different types of affinity reagents, whether you're profiling for RNA species or protein species. But these are essentially standard probes that are standard types of reagents that are used to profile tissue, whether it's an ISH probe or an antibody, that have been barcoded with a particular oligonucleotide that we know the specific sequence for. That barcoded sequence is attached with a UV photocleavable linker, and that's how we specifically allow you to capture which molecules are present within a given part of the tissue. And then the last component that's really critical is that we array a series of micromirrors that allow you to tune where you shine light. So the GeoMx platform is by definition a microscope. Microscopes do a great job of illuminating tissue, and this micromirror array allows us to specifically control light down to a micrometer. So by having this combination of both a multiplex IF, a micromirror, and these affinity reagents, we can now start to profile specific regions of interest within the tissue. Now, within the lymph node, we purchased a series of blocks that were from inflamed lymph node from patients that had been harvested without any complications or disease that has been noted. Specifically. And what we wanted to do was look throughout the tissue and identify key structures that we could start to profile and understand differently. I want to make sure that you understand that there's going to be a couple different experimental designs that we're pursuing across these different samples. The first one is to try to understand how T cells and B cells are particularly impacted as they move throughout both the cortex and the medulla. We have regions that are T cell rich zones that are collected in the cortex, as well as regions that we've segmented where we look for, where we draw a square polygon in the middle of the medulla here. And we're segmenting using the immunofluorescence staining that we have to collect specific cell populations. Now, I'm going to talk through a number of these findings, and one thing that I want to mention is that all the data that's being collected for this is available on our website. So if you want to explore this more, I'll show you how to do that later on in the talk. So just to emphasize again, the types of regions that we're collecting here, we've got 3 different regions that we're just hand drawing the shape, and we're going to collect it specifically from the tissue. We've got regions that are T cell rich and B cell rich, and we also have regions that should be heavy in stromal cells, and these are called trabecula. We also, as I mentioned, place several square regions within the medulla and have segmented those into T cells, fibroblasts, and B cells simply based on the staining. And what you can see, the difference in terms of how these are built, the ones collected at the top collect every single cell within a region, regardless of its cell type. The ones that are from the segmented profiling are going to be focused really on the cells that specifically express a particular immunofluorescent marker. Now, we wanted to go through and confirm that what we're collecting makes sense biologically and can help us drive how we look at comparing these different regions of interest. The first way that we did this was comparing our tissues to the Human Protein Atlas. The Human Protein Atlas is a compendia of tissues that have been collected over time and which have been profiled with as many antibodies as possible. And so we can test whether or not we're seeing expected staining patterns within our tissue. I'm going to focus on the segmented profiling because those are regions where we should expect to have really strong composition of individual cell types. So the first example was that to go through and look at CD3 staining. Now, CD3 and CD8 are both markers of T cells, and we should expect to see those enriched specifically within the CD3 compartment that we specifically collected. Here, what we're observing is staining that's shown from the Human Protein Atlas, and in the middle is the CD3 expression from our particular experiment, where you can see that the CD20 and SMA positive regions have very low staining of CD3. While we do get specific collection of CD3 itself. The same is true for CD8, which is a marker of activated T cells. We also went through and collected B cell rich regions. So we're showing similar plots as has been shown here to the previous one, where we were looking at T cells. And so this again confirms that the collection that we've got is both specific and sensitive to the markers that we anticipate to see in these regions. Finally, the SMA-specific regions are regions that should have high degrees of fibroblasts and cells that are adjacent to those fibroblasts and make up structural components within the tissue. And what we can see here again is that SMA is specific, but we also get genes like aquaporin, which should be interacting with the stromal cells and may be more highly present within that type of tissue structure. All of this is really important to make sure that we understand that the system has been performing as expected. However, none of these are particularly striking or novel findings, but what this has enabled us to do is to feel confident that when we do see something within our study, it's likely going to be driven by real biology. And the Human Protein Atlas is a great place to validate any of the findings that you have if you're working on our system. The benefit of using this as a validation source is that it is the functional readout as a post-translational readout, while we are also profiling just RNA species expression. Now, as I mentioned, we're profiling RNA, and so we're really trying to get at not just any one individual marker, we're actually profiling the whole transcriptome at once. And so we can now start to ask questions as to what changes when you want to compare different regions that are enriched with certain cell types. Here we're looking at, on the left-hand side of the volcano plot, we're looking at which genes are enriched when you're in the B cell zones that we collected, which were highly focused around regions that were rich for B cells themselves, but not specifically segmented on them. And on the right, you're looking at T cell enriched zones. Now, as before, we're seeing specific markers that we know to be expressed by B cells or by T cells, such as CD20, CD19, CD79A should all be directly expressed by B cells. But we're also picking up markers of genes that help us understand how the cells are polarized and how they move throughout the tissue. So, for example, CD3, the CD3 rich T cell rich regions also show examples of expression of markers like CCL19 and CCL21, which are genes that are specifically involved with chemotaxis of T cells themselves, and are known to specifically be enriched within the lymphatic system because they're involved with T cell maturation during the process of antigen presentation. So I want to make sure that what's emphasized here is that while we can specifically look for regions that are defined by immunofluorescent markers, what we get is a snapshot not just of those genes and of those particular markers that we know should be there, but a deep dive into all of the particular expression patterns that may underlie the functional biology that we're looking into. Now, this is an example of the T cell versus B cell zone. I also wanted to show what the data looks like for the stromal rich regions when you compare, for example, the CD3 plus cells versus the SMA cells. And again, as we mentioned, we do see enrichment of genes such as CD3, CTLA4, but we also see aquaporin, PKM1, and a number of markers that are specific to extracellular matrix showing up within the regions collected that were enriched for SMA. All of this data is really interesting in that you can start to peer through this and identify novel particular genes that you may not have identified before from bulk or single cell profiling. But it does also allows us to take a next step further into understanding what this tissue functionally does. When we're looking at our expression patterns here, one of the common ways in which analyzing data from either bulk or single cell profiling has been performed is to contextualize gene sets by comparing them to previously annotated gene set ontologies. And so we've performed a similar analysis here. We've used tools that have been developed specifically for RNA-seq and single-cell-seq to do this, but they're completely appropriate to use with our data type as well here. And the one that we're going to use first is going to be using single sample gene set enrichment analysis. What this is going to do for us is now we can take a look at every region that we've profiled and ask the question, which pathways are particularly enriched within that region? We can look at the genes' relative expression and abundance within our tissue, and use this to score the genes as a set themselves. What you can see within this particular graph is that there's a number of genes that make, or a number of pathways that make particular sense for each particular structure, and we do see enrichment and specificity for these structures. Each of these pathways was identified as being differentially enriched within a given type of region that was collected at an FDR of less than 0.05. And so you can start to look through this list and see some really interesting biology that may not have been able to fall out at the individual gene level, but might be more apparent when you're looking through a gene set ontology here, whether that's how the tissue would respond to a viral infection, whether that's maturation of B cells or CD28 cross-ligation. I want to emphasize that all of this is entirely agnostic to, we did not set up a model to specifically assume that any of these gene sets were enriched. All we did was ask which gene sets are differential between these different structures. One thing I noted at the onset was that we did this type of study across multiple different blocks. We actually purchased 5 different patient samples, and we're able to run this study across not just one sample, which is what's being shown here, where we're collecting 5 or 6 regions per particular structure. We also were able to do this across the entire cohort. And again, any of these types of analyses are robust to being able to look across cohorts. And the benefit of this is that it allows you to feel confident that the results that you're getting aren't driven by a single given patient. When we're doing the analysis using a single sample, we can use tests that are usual within the statistics space for most biological applications. You can use things like ANOVAs or t-tests. When we move to a space where we're looking at replicated samples within a given patient, we do move to more advanced models, and we have documentation on how to do that within our systems, both on our website, where we discuss some of the analysis, as well as on Bioconductor, where we have several packages for data analysis directly for GeoMx. If you'd like to learn more about that, feel free to reach out to us. So this is one way of looking at the tissue, but as you can imagine, there's multiple different regions within the tissue that are particularly important. One of the things that we didn't emphasize a whole lot within this was the structure that is where a lot of the immunological action happens in terms of where cells mature and how the body filters through and looks for antigens that are specific to viruses and bacteria. So in addition to the previous profiling structure, we also undertook a profiling where we're looking specifically at the germinal center within the tissue. This is looking at the gross level of overall what's happening within the germinal center as a whole. And I'll come back to further refining this later on in the talk, because this is a very high view of what's happening in a germinal center, but we can take even further granular cuts within the structure. Here we're showing an example of a tissue where we've got germinal centers that are these round structures where we go in and profile those round structures and then segment them further. But we're also collecting mantle zone B cells, which are B cells that live adjacent to the germinal center, as well as T cell rich zones that are adjacent to the germinal center as well, so that we can understand some of the changes that happen between and across these different tissues, between and across these different structures. So I want to show what's happening within this segmentation because we did a couple specific tricks here that we wanted to make sure you understood. The germinal center is composed of a large number of cell types and is basically the meeting ground where everybody comes together and decides what the plan of action should be when faced with a new antigen. It's composed of, as I said, several different cell types, but we're going to focus on 3 of the ones that really do a lot of the work and heavy lifting within the germinal center in terms of setting up an immune response. The first of these is myeloid cells that are actively presenting antigens and helping T cells understand what they have observed out within the wide world of the body. We're marking these cells with CD11C, and CD11C marks primarily dendritic cells. It may also capture some other myeloid cells that are in different stages of differentiation. The next cell type is the B cell. And again, this is another antigen presenting cell. It's also a memory cell that helps the body understand what long-term pieces of information it's seen in the past. And so the combination of these two cell types really helps T cells understand what they should be attacking and what they shouldn't be attacking. And so the final cell type that we have profiled here is the T cell, which is CD3 positive. So these three cell types are the ones that we're going to focus on here. And we're going to look again for differences that we observe between them and across them. So the first, again, is to prove to ourselves that we do feel like we're getting strong and specific staining of the different markers. When we looked previously at CD3 and CD8, we saw that both of those were enriched within CD3 positive segments. One thing that is different here, and I want to highlight, is that germinal centers don't typically have a lot of CD8 positivity. And this is, again, something that we can see directly within the Human Protein Atlas. And it's also something that we observe on the right-hand bar plot here. CD8 expression is differentially enriched within the CD3 positive cells, but it's not very high and not very significantly separated from the other compartments. This is simply because we don't see a lot of CD8 positive cells within the germinal center itself, as they're not the ones that are really doing the business end of trying to identify whether or not an antigen has been presented, seen, and is worth attacking. They get mobilized in the adjacent regions to the germinal center itself. Similarly, when we're working through understanding the biology of which cell types from the myeloid lineage might be present, we also see direct evidence that CD11C is staining specifically as we would expect. Its protein marker is called, or its RNA gene symbol is called ITGAX, but the protein name is CD11C. So when you're looking these up in the Human Protein Atlas, this is how you would find it. Similarly, myeloid cells and macrophages are commonly marked by a gene called CD68, and we see specific enrichment of that with the CD11C compartment, suggesting that these are in fact myeloid cells that we are picking up, and that there may be some macrophages also within this particular compartment of the tissue. Now, I wanted to give you one example here, again, of the type of work that we can do when we're trying to compare different cell types. Here we're looking at CD3 versus CD11C to see what's happening within this structure of the tissue. One thing that is obvious and easy to note here is that, well, previously we had seen that CCL19 and CCL21 were being enriched within the T cell zones that we had previously profiled in the germinal center. That's not a function that is really being performed by the T cells themselves. CCL19 is sort of sitting within the middle of the volcano plot here. That's because these cells have already migrated in. They don't need to express it, and they're not actively pushing that particular axis. However, what we do see is strong enrichment of genes like CTLA4, as well as genes such as IL-18 within the dendritic cell compartment. And both of these are really critical in terms of, again, making sure that the right cell types are present, as well as making sure that the T cells that are present within a region are not going overly active. So CTLA4 was a gene that was identified previously. It was actually discovered really within the lymph node and has played a huge role in terms of both immune cell development as well as overexpression of that particular markers is a target now in cancer therapeutics and immunomodulation within autoimmune disease as well. We're able to directly detect this within a single sample of this tissue. Now, I want to point out, I've not been able to dive through all the data that we have here. And the reason that I'm not going to go through every single piece of data that we have within this study is because we've made this publicly available on our website. If you'd like to download the data or explore it, we have interactive stories that allow you to explore the lymph node directly on a page called the Spatial Organ Atlas. So if you want to scan the QR code and thumb through that while we go through the rest of the results, you absolutely can. There's multiple different organs that you can view, and the lymph node is one of 5 human organs that's on that page. We also have developing mouse and the mouse brain as well on that page. These stories allow you to go through and look at the experimental design that we've set up. They walk you through QC of the data and give you information about how many genes we're detecting within specific regions. And then we also allow you to explore some of the high-level results that are found when comparing different regions against each other. And the benefit of using this is that you also get the chance to view the tissue itself, the segmentation strategies that have been performed, and all of the immunofluorescence staining that has been captured within the experiment. So I would highly recommend that if you get a chance to take a look through these data, because we've got plenty more results available there, and you can download and work with the data on your own directly from the website as well. We make sure that all the data that's related to these experiments is present, including the raw count data that's generated, as well as data that has been through the normalization process, and all of the raw images that have been captured from our system, so that you can take those into your favorite image viewer or editor and then move forward with that. So I want to highlight a couple things as we move this talk forward about CosMx. Before I jump into that, we did get a question about immunostaining. And I want to go ahead and grab this right now while we're talking about GeoMx, because I think that I understand the question, but I may need a little bit of clarification here. Essentially, the question is, if target proteins I want to analyze happen to co-localize after immunostaining is done, would the DSP be able to segment and effectively collect separate samples for posterior transcriptomics analysis? So after you've done the immunostaining, you have the ability to control the segmentation process however you want. And by that, I mean you can segment on multiple different markers or on individual markers. So if you want to, say, look at a tumor that has T cells that are infiltrating through it, like is shown at the bottom of this figure below, you can collect tumor cell rich regions using pan-CK, but then use CD3 staining to exclude the T cells that have invaded, so that you can capture those in a separate segment. Or you can capture them all together as just a tumor compartment. But that control is totally up to you. And so I believe that that is the question that you're after, but if not, please feel free to further clarify it in the comments as well. Now, as we move over to the CosMx side of the house, I want to emphasize that, we've had a lot of success and a lot of interest in understanding the overall tissue structures and cellular dynamics that are happening at broad scales, but sometimes you really need to understand what's happening when 2 cell types interact with each other in local regions of the tissue. As I mentioned before, the way that we set up the first Germinal Center experiment was rather broad. We took the whole Germinal Center, and we didn't have a lot of granularity as to what was happening within different regions of the tissue. Here, CosMx allows us to ask questions at the cellular level of what cells are interacting with each other and what particular marker genes might be going together within different regions of the tissue. I want to talk a little bit about the chemistry of CosMx so that you can understand how it's similar to GeoMx and where it differs, because we use some of the same tricks as we did before. So the first piece of information here is that as with GeoMx, you're working off of a standard histological slide. And again, you're using affinity reagents to understand what's happening within your tissue. As with GeoMx, Within CosMx, we only adhere the affinity reagents once. You never strip them off of your tissue, and they consist of standard probes similar to ISH probes or antibodies that you would use in other applications. The magic really happens, again, by the fact that we're using those probes, those particular affinity reagents, with barcoded sequences that we can read out. But instead of collecting the sequences, like we do with GeoMx, what we're doing is going to directly image them within the tissue. And to get the imaging to work right, we have to do a few tricks. And again, we rely on a couple different facets of our reagents to really drive forward how we can then measure them specifically in space. The first of these is that instead of having a barcode that we detach from our RNA species or from our antibody, all of that's physically conjugated together, so it's all one molecule and is never going to break. Second, what we do is we use a secondary oligo to bind to that. We have regions within the tag that is not homologous to the RNA or the tag that hangs off the antibody, which we call landing pads, where an additional species of molecule can bind to it directly. This species is the one that's conjugated to a fluorophore series so that you can specifically image whether or not a binding event has happened. After a binding event has happened, what we'll do is a round of imaging where we'll look for different colors of fluorescent molecules showing up specifically within our tissue. After we've done that one round, we now use UV light, again, like we do within GeoMx, to specifically release the oligos using that photocleavable linker. We wash the tissue with a very light wash that removes any of those fluorescently tagged oligos and allows space for new molecules to bind. And so over a series of cycles where we do this flowing on of fluorescently tagged molecules, imaging, hitting it with the UV light, and then removing those fluorescently tagged molecules, we get a series of sequences that helps us identify where specific molecules have been bound. What this produces is a data set that's extremely rich and looks something like this. What you're going to be able to have when you do a CosMx experiment is an understanding of where individual markers of individual genes are. So every point within this graph represents a particular RNA transcript that we are confidently detecting within our tissue. The lines around them are the cell boundaries. As I mentioned earlier, CosMx is collecting, just like GeoMx, not just the location of any individual ISH probe, but also multiplexed immunofluorescence. As you can imagine, this is generating a ton of data. I just want to emphasize that if you're running a whole tissue sample, you're generating millions of cells and potentially billions of transcripts within a single run. And so I'll come back to that a little bit later, but we want to make sure that that doesn't become a barrier for analysis downstream. Let's talk a little bit about the different layers of data and how you can use this to understand your tissue. This is, again, not lymph node. It's just for the specifics of understanding what you can do with CosMx data. But we've got 1,000-plex RNA, and in R&D, we've been doing some work up to 100-plex protein. I believe at launch, it's going to be closer to around 70. But what this is going to provide you is within a given particular sample that you're looking at, you're going to have at least a 4-channel multiplex IF, RNA expression that goes with that at the ISH level of up to 1,000 plex RNA. And then you can perform downstream analysis that allows you to identify the segmentation of your given cell, as well as perform analysis to identify what cell types are present within your tissue, what their activation state is, and how they interact with each other. So all of this is generic, so let's dive into a little bit of the lymph node biology. As I mentioned before we had been looking at the Germinal Center as a functional unit that was indiscrete. We were looking at the whole thing all at once. That's not how the Germinal Center really functions. And so we wanted to see if there were any of our samples that had really well-defined germinal centers. And when you say a well-defined germinal center, histologically, that's looking for a germinal center that has both a light zone and a dark zone. And the light zone is where the T cells and the B cells really mature together, and they go out from the germinal center. The dark zone is where B cells specifically proliferate and try to help produce a mass of specific B cells that are going to be specific to antigens that might be presented down the road. This is where B cells perform hypermutation on their particular B cell receptor, so that you have a diversity of potential antibody producing cells. It's when they move into the light zone that they become specific. And so what we found was there was one particular lymph node within our study that had a large number of germinal centers where we could identify the light zone and the dark zone by using staining for Ki67, CD20, and CD3. And in this particular sample, what we did was we collected every lymph node that we could by GeoMx, segregating those into the light and the dark zone, as is sort of shown on this graph on the right, but also capturing an intermediate phenotype, knowing that we might be seeing more specific signal if we separate the light and the dark zone more consistently from each other. And we might capture biology that is only present at the interface between these two zones if we do separate them into three rather than just two zones. We also took this same sample and then profiled it with CosMx. We used our R&D kits for testing for 1,000 subcellular transcripts as well as 100 proteins. And this translated down then to 27 cell types that we were able to profile across the entire tissue structure. And here we're just showing example expression of 6 of the different transcripts that are present within the tissue. We'll dive more deeply into some of the other results that we're finding here. But this combination of these two studies really starts to now allow us to get really fine-grained understanding of what's happening within the lymph node germinal center itself. Now, I say this, but I want to really emphasize that one thing that we spend a fair bit of time that we do with our systems is making sure that we're getting robust data. And so I showed you that previous slide that showed the lymph node sample as individual genes. This is showing it on the left as cell types, colored differently by the cell type that's assigned. But it also, as you can see in this particular blow up in the middle, you can capture a light and a dark zone of the germinal center, even just by looking at the cell types that are enriched within that spheroid structure. But it's really critical to go down to the segmentation level to make sure that you've got the right cell shape to be able to call these cells appropriately. I want to emphasize this simply because it's where we've spent a fair bit of time. We had a poster that we released at AGBT that described our methods for cell segmentation. And the big thing that I want to point out here is that we're not just using nuclei segmentation, we're using a consensus model that involves both nuclei segmentation as well as cytoplasmic segmentation to create the final cell segmentation model that we work with. We've done this within the lymph node sample that we profiled to make sure that if there's cells that are irregularly shaped, that we can better profile them. If you'd like to learn more about how we perform this segmentation, there's a poster from AGBT on our website that we're happy to point you to. Now, I also want to emphasize that the other piece of this, when you've got a tissue structure like this, you can basically get the cell types for free, but the reason that you get the cell types for free is because we're working in a space similar to what you have with single-cell-seq. You have data that is physically resolved, but you also have data that's coming directly from transcriptomics, and so you can run the same types of analyses that you would with single-cell sequencing. So here on the right-hand side is showing the UMAP projection of these cells and their relatedness to each other, regardless of their physical colocalization, which is shown on the left. And again, as we have this in hand, we use this directly for doing our cell typing. This helps us identify cells that are specifically related to each other and which should be biologically similar regardless of their physical location in the tissue. To prove that we've done a cell typing that's robust and useful, we take this the next step further of going through and comparing the cell types that we've identified to the genes that are most specific to these cell types. And so this is one of the QCs that we perform in pretty much every experiment that we run, because we want to make sure that we see the appropriate cell type markers in specific types of cells. For example, you'll see within the middle of the graph, there's this block of red that is associated with CD3, CD8, ITK, and a couple other markers that we know should be expressed across a broad range of T cells that exist within the lymph node. Conversely, we've also got markers that are almost entirely specific. And so we're able to pick up things like VWF as being very specific for endothelial cells within this system. So I want to emphasize that this is a QC that we run before we do any downstream analysis, because we want to make sure that the cell typing that we've performed here is robust and captures most of the data that we're looking for. Now, I mentioned that we collected all the germinal centers here with GeoMx, so what I'm going to talk through over the next couple of slides is how we approach this experiment of taking not just CosMx data, but GeoMx data as well, and integrating that together, and what stories we can find by doing so. So here we're going back to the GeoMx data, and this graph essentially is a three-dimensional volcano plot, where we're looking for enrichment of individual pathways in either the light zone, the dark zone, or in the zone that sits within the middle of them. Now, one thing I want to emphasize here is that if there was no novel biology in the intermediate zone, that area would be mostly, if not entirely, absent from this graph. But for the purposes of what we're seeing here, in terms of understanding specific biology for the light and dark zones, we can find different pathways that we can then map back to and physically explore within the tissue. One example of those is CD28 co-stimulation, which we then show on the right of the scores for that particular pathway being graphed physically on the tissue in those breakout graphs below. And so anything that's enriched in yellow is being enriched within the light zone. Anything that's shown in dark blue is not really expressed. And you can see a direct gradient from dark zone to light zone of the CD28 co-stimulation pathway. This is great for understanding the overall functional consequence that's happening within a given tissue structure, but it doesn't tell us what cell types are actively driving that interaction. CD28 is extremely well characterized, and we have some pretty good guesses as to what cell types are going to drive that. But in addition to looking at the overall pathway, we can also do what we call a ligand receptor analysis that's spatially resolved. We can look for which cell types within a germinal center are specifically interacting with each other based on their proximity and expression of ligands and receptors on those cell types. And so here we're using the physical location to drive our understanding of which signaling cascades are being driven by which cell types. And so for the example of CD28, we can see that CD28 cross-ligation is primarily happening within the germinal center T follicular helper cells, and it's primarily being driven by its interaction with those light zone B cells. This is one of several different interactions. There's others in this that make perfect sense for the biology that we're capturing. For example, ICOS is known to be upregulated on T follicular helper cells and was identified as a major driver of maturation of those cells within the germinal center. We see that here as well. But as before, we can use the CosMx data to identify the players that are really driving an interaction. And then what's shown here on the right is the downstream consequence. So again, when we had previously shown that the CD28 co-stimulation pathway is enriched within the light zone on this graph, we can see all of the different genes that are specifically involved with that within the GeoMx data as well, if we look at it as a heat map, where here we're seeing enrichment of CD28 co-stimulation being driven by all the genes that are shown on the right. Now, the last piece that I want to emphasize is that sometimes it's really important to drive understanding of biology, not just at the RNA level. As I mentioned, within this particular lymph node sample, we also have protein staining that we did with the CosMx platform as well. And one particular marker that was of interest that we saw in previous work within the GeoMx data was that IL-18 seemed to be of particular interest within the myeloid cell lineage. And here we see evidence that IL-18 is also expressed on the myeloid lineage, and it's likely talking to B cells within the germinal center. So we wanted to take this the next step further and understand, do we see evidence of localization of IL-18? Because cytokines like IL-18, the interleukin family especially, is highly post-translationally modified and regulated. They need to be actively maintained, and they need to be loaded into granules sometimes or bound to the cell membrane. And so RNA expression here may not be sufficient for proving that this axis really is active within our sample. So we looked within the CosMx protein data, and we asked, where do we see IL-18? And so this animation shows you a little bit about which cells are expressing IL-18. These are going to be the green arrows, are myeloid cells that are expressing IL-18. Now, I want to point out that IL-18 is expressed by other cell types, but when you look for which cell types within the germinal center, that round structure shown on the right, are expressing IL-18, it's almost exclusively myeloid cells. And so we believe that this is indicating that this is a novel function for IL-18 within the germinal center itself that may have previously been underappreciated. So with that, I hope I've given you some insights into some of the ways in which we're exploring the lymph node right now. This data is actively being analyzed, and we're looking forward to seeing more findings like this become validated. But we're hoping that this type of data can show you how to use these two systems together. And so I'm going to end here on this slide where we emphasize again, you can use GeoMx to really do high depth profiling of the functional consequences of different structures within your tissue, and the interactions that happen between different cell types at their functional state level. And CosMx can really help you understand which cell types are interacting with each other and how they interplay at a very deep level. This slide also emphasizes that as these data are being collected, we recognize that there's a large amount of data that's being produced by both of these platforms, and studies that are going to be translationally driven are going to be ones that produce larger and larger cohorts of data. So we have also been in the process of launching a platform called Atomics, and this is where we will create a data hub for all of the data collected from both of our systems, and we'll be able to analyze and integrate this data as you work with it going forward. If you'd like to learn more about Atomics, go to our website, but I wanted to make sure that that was something that you were aware of as well. So with that, I think we've got a couple minutes to jump into questions. And so thank you all so much for attending today.
Thanks so much, Jason. Great informative presentations. We had a few questions that came through. One of the questions that came through is, can you do spatial transcriptomic and proteomic profiling with GeoMx from the same tissue section?
Yeah, that's a great question. So as I mentioned before, our protein profiling at minimum is you get at minimum 4-color immunofluorescence. However, we recognize that some people have very, very precious resources in terms of samples available to them. So we launched a proteogenomics workflow where you run both of our assays, the protein and the RNA assay, directly on the same slide earlier this year. You can find a white paper related to this on our website, and there'll be more information coming. But this is absolutely possible now with GeoMx to just use a single slide for all of your analysis.
Yeah, and I will add to that question that came in. We have a webinar that's happening next week on Thursday, October 27th, a week from today, that's jointly sponsored by Illumina. And there's researcher Sudarshan Damodaran at University of Wisconsin-Madison. He's going to be talking about using this spatial proteogenomics workflow to profile the whole human transcriptome, as well as about 150 plex human proteins with GeoMx in different tissue structures within diffuse midline glioma samples from pediatric and adult patients. So if you're interested in that, you can go to our website to the events page and then click on the webinar that's happening on October 27th and register for that. And we have timings both for Asia Pacific as well as Americas and Europe. So another question that came through, is the tissue destroyed after immunohistochemistry or molecular profiling? And I guess this question could be asked for both CosMx and GeoMx. And in other words, can you perform an H&E or chromogenic IHC?
Yeah, that's a great question. So the way that GeoMx works, the only thing that you're doing with GeoMx is a standard histoprep, and you're only touching the slide with light. All you're doing is collecting things that have physically floated off of it. So GeoMx is by nature non-destructive, and you can work with the tissue afterwards. The flow cell that's attached in CosMx is a little bit beefier, and so it, I believe, is still non-destructive. You're not physically doing anything to the tissue, but it may be harder to remove the flow cell for CosMx and work with it downstream. But we have, for GeoMx, had plenty of people work with our slides after the fact and do repeated stainings or run another assay that they wanted to, even with GeoMx as well, if they decided that they wanted to expand on a previous protein panel and use a complementary one that they didn't run the first time.
Excellent. Thank you so much, Jason. Another question that came in, do you have a list of custom morphology markers or stains that have been used with the GeoMx digital spatial profiler?
Yep, all that information is on our website. And so if you want to see what our technology access lab has been doing across different tissue types and across different morphology markers, I'd recommend you go take a look on our website because that lab has worked with many different types of tissues and is great for getting a sense as to which antibodies have worked really well in those tissues. So we collect that information and make it available directly to you.
Thank you so much, Jason. Another question that came in, is it possible to use the CosMx spatial molecular imager on tissues other than human?
The answer to that's absolutely. So at launch, we're producing both human content and content for the mouse. One thing that I will note too is that just because a panel has been built specifically for one species doesn't necessarily mean that you need to feel like you're limited just to that species. We have had people use the human CosMx panel with non-human primate already in studying the response to the simian virus that is similar to HIV, the SIV virus. So we have seen people use these panels in new and inventive ways that we did not develop them specifically for, but which we can help you understand as you go forward. Similarly for GeoMx, if there's a particular species that you're working with, you may well be able to work with it with the panels that we already have available. Last week, I saw someone present research using the mouse panel with hamster. And so I want to make sure that you understand that there's lots of opportunities, both in terms of us producing panels that have directed content for a specific species, but also for potentially using the panels that we have for cross-species analysis as well.
Excellent. Thank you so much, Jason. So we have just a minute or two for any additional questions. We've gone through all the questions that were submitted during the course of the webinar presentation, but if there are any of you that have any burning unanswered questions that you'd like to get out, now's your chance. So I'll give a minute or two if you want to type in your questions. I also want to mention that this presentation will be available on demand after the fact today. So if you have any colleagues who were planning on watching this but weren't able to attend, or if you have colleagues that you think might be interested in this presentation, please go ahead and forward them the link to the registration page. All right, I think that concludes the Q&A portion of the webinar. I want to thank everyone for joining us to listen to this presentation. And again, I'd like to thank you, Jason, for joining us as well.
It was great talking to you all today. I hope that this has been informative for everyone, and I hope you have a lovely afternoon or morning, depending on what time zone you're in.
Great. Thanks, everyone. Bye-bye.